Hello, welcome to my Stonewall herp species experiment by me, Simcha. I'm going to start with the importance of stonewalls as an introduction. Stonewalls are important because they provide unique habitat for many species, including herp species, such as the ones in these images, American bullfrog, little garter snake poking its head out from under a rock, and some redback salamanders. According to Thorson, 2009, amphibians are surprisingly common around stone walls, and reptiles are furtive dwellers in stone walls. So it is very well known that around stone walls there are herp species. So what are stone walls? Stone walls are fragments of the earth crust built into a wall for human use. These stone walls provide habitat for a variety of life, and as you can see here, they run along a length of some distance and tend to be stacked in a particular way that designates it that you know it's a stone wall. Stone walls built without concrete are called dry stone walls in Europe, such as in Italy and across all of Europe. So why study stone walls? There are actually very few studies about the relation of stone walls and herp species in terms of diversity or abundance. From the studies that are available, they indicate that there is an increased abundance of herp species on or near stone walls. The overall density of these herp species near stone walls tend to be higher than densities in other areas, and stone walls near ponds have an even greater abundance of herp. And you can kind of see in this image how there's also areas around it that herps could be living in. So what habitat surrounds stone walls? In my study area, Northwoods, the habitat is mixed oak and pine forests with some vernal pools and wetlands. Most of the stone walls were in the oak-dominated areas, as well as some near vernal pools or other forest wetlands. You can kind of see that these are definitely oak trees with all the oak leaves on the ground. But there were some limited amounts of stone walls also a little bit in the pine areas. So what herp species are likely to be found in Northwoods? There are a variety of herps all across New England and in Rhode Island, but for more forested areas and potentially near stone walls, some examples are redback salamanders, American bullfrogs, wood frogs, pickerel frogs, American toads, eastern newts, common garter snakes, common ribbon snakes, Decay's brown snakes, eastern milk snakes, and eastern hognose snakes. As you can see in my images, some bullfrogs are found, redback salamanders are found, and garter snakes are found primarily, but it is always possible to find other things around in that area. So, my main question, how does proximity to stone walls influence diversity or abundance of herp species? In terms of materials that I use while herping, I mainly use my iPhone that I was able to take pictures with included metadata to note down observations. So I was always able to put the species, the count, the location, the trail, and the proximity of stone wall, as well as any other extra notes. And the metadata from the image was able to show me the day, the time, and even a little location. And I also had myself wearing weather-appropriate clothing. So my study area, Northwoods. This is the Northwoods trail map, and I selected this as my study site. There are several stone walls, notably near the yellow trail and the parts of blue trail south of that crossroads entrance here. There are also some pretty far from the stone walls, such as along a lot of the driftway trail here. So my procedure. I would follow marked trails to guide my path transect when exploring area, but then I would go off trail and lift up rocks and logs that can be safely accessed and lifted or rolled. When a herp is found, I try to catch it, and there we go, have it in my hand. Once I caught, I try to contain it. For salamanders, I tend to cut my hands around it to calm them down. And then I take out my phone and I get some pictures of it, typically at 1x and 2x zoom. And then I also record the observation on the note in my phone, which also is what was seen in the previous image of what I recorded down. And then I would put the herp back where it came from. And as you can see, I was able to catch some garter snakes and a bunch of redback salamanders. I wasn't able to catch the bullfrogs, but I was able to still get pictures from a distance. So if I can't actually catch it, I try to at least get a picture from a distance. So here are my graph of my results. As you can see, I found a lot of redback salamanders. And there was a pretty decent amount that were on the stone wall or very close to a stone wall, and even just relatively close. And also then there were a bunch that were quite far away, but overall there was a lot of abundance near stone walls. And then to make it easier to see the smaller bars here, when I exclude redback salamanders, you can see that there were bullfrogs and garter snakes and wood frogs that were also found, and some caught, with a range for the garter snakes, but the bullfrogs and wood frogs were at the vernal pool, which was considered relatively far and within visual distance. So my overall herp species and abundance found: redback salamanders, I found 166 individuals, but over 167 observations, as I had one repeat observation. I found 7 American bullfrogs, with one of them being found multiple times across multiple days. So I only counted that as 1, and then when I found more of them, that's when I was able to find a total of 7. I found 6 garter snakes. I was only able to catch 5 of them. And wood frogs, I found 2 of them, but wasn't able to catch any of them. And here's some more pictures of redback salamanders and the bullfrogs, 2 in this picture, and a garter snake. That was actually a pretty large one. So, conclusions. There were relatively high abundance but low diversity of herp species near stone walls. As you could kind of tell, I only found 4 different species, but very high abundance of the redbacks. Redback salamanders were generally overall abundant, with a lot present near stone walls, especially near the vernal pool. These findings align with previous studies that have noted higher abundance of herp species on or near stone walls, especially near a pond. And then these are some more images of redback salamanders, and these are just a few of them. I have so many images of redback salamanders. As for limitations on this study, in overall data collection, there was a massive snowstorm that made it so that there was snowpack still on the ground until about mid-March. So I couldn't start collecting data until then. So that pushed a lot of data collection till later. I also didn't have all the time in the world, so I had a lack of time availability on certain days within the data collection period that would have been great to go out, but I just wasn't able to. Then while actually herping, there were some inaccessible areas due to high density of bramble pricker bushes, kind of like this one here, and so that means that I couldn't safely access that area to lift the rocks or logs. And then also my own strength, because I was working alone, lifting the rocks and logs had to be within my capabilities. And the end. Thank you so much for watching.